Hello students, welcome to Biosastra Talks. I hope all of you are doing well. As we have went with the part 1 of questions from 151 to 180, 151 to 180. In this video, we are going to learn about the remaining questions and the last questions so that we can end the paper solving the paper code 45 from the biology session. Okay, what was the first question is role of the water vascular system in the echinoderms is. So in this question they are asking about we have to say what is the role of the water vascular system that is present in the echinoderm. So what were the options they have given is first one is respiration and locomotion, second one excretion and locomotion, third one capture and transfer of food, fourth one digestion and respiration, and last one digestion and excretion. So in these options you have to say that water vascular system of the echinoderms are mainly they'll play major role in which of the metabolic process. Okay, you have to see here if you see that this is an echinoderm. Normally if you see here this is an echinoderm, like it is a starfish. Actually starfish is also an echinoderm, right? So this is the picture of a starfish. Here you can see that there are bubble like structures like balloons like so they are known as the water tubules, and that the connecting line between these bubbles is known as radial canal through which the water will flow through the entire body of the starfish. Okay, this radial canal is connected to the ring canal, like that it can supply, this ring canal can supply to the remaining parts of the supply water to the remaining parts of the starfish. Okay, so here you can see that starfish is having 5 or 6 arms, like that 5 arms. So each one will have different, like we are having the digestive system, nervous system, like that. In the starfish you can see that one arm is having a digestive glands, another arm is having nervous glands, like that. So here in one arm you can see the water vascular system through which the water is supplied to the whole entire body of the starfish. Okay, so here you have to say what is the main role of this normally water vascular system is. First thing is normally to with the help of this water vascular system in only the starfish can move. Okay, so it helps in the locomotion, and second thing, it also means a starfish also respires through this water vascular system only. So normally you can say that the directive feature is normally it helps in the capture of the things which it have to take inside, like the praise. Okay, and the transport of food from one place to another. Why? Because we know that vascular system in the plants also, like xylem and phloem, what they'll do, they'll transfer the water and food materials throughout the body. Like that only in humans we'll have the blood vascular system. Like that only in the starfish also you will have this water vascular system which will supply the transport other, like which will supply the food throughout its body. So here it is helpful for capture and the transfer of food and locomotion and sometimes respiration also. So here you can see what are the main process that were that were done with the help of the water vascular system or respiration, locomotion, capture and transport of food. Okay, so here A and C are correct. The option which is having A and C is second one, so option 2 is correct. If we move on to the next question, what is the next question is here this question came from the immune system. Actually what they are asking is from this question, which of the following type of immunity is present at the time of birth. Okay, here you have to say that we are having so many type of immune responses. In that you have to say that at the time of birth which type of immunity is seen in the fetus or the baby or child. Okay, and it is a non-specific type of defense in the human body, and it is also called as non-specific type of defense in the human body. Okay, so normally you can simply say that specificity means we'll concentrate on any one thing. Like non-specificity means it will not concentrate on anything. Means non-specific type of immunity matlab means normally what will happen means they'll not target the specific cells. Okay, so this immunity is normally, you can say that it is a basic immunity. Normally we know that the babies will have the basic immunity, they cannot develop the whole immunity, right? Why? Because for example, when we'll grow adults, like when we have any fever like dengue fever or something, what will happen? Our body will produce antigens against that dengue fever. But in the childhood, they don't have any like infections or something. What they'll do in the childhood itself, in the birth, at the time of birth, the baby will have the normal immunity, that is the basic immunity, which is non-specific, means it will have common antigens to fight for some fever, like that, fever, cough, cold, like that. So that immunity is known as innate immunity. Okay, so it is present in the adults also. In the adults, we'll call them as non-specific type of defense, means it will show non-specific type. You cannot say that it will definitely fight with this type of like dengue cells or malaria cells. It is non-specific, means it can fight when any host or the parasite enters. Okay, normally non innate immunity or non-specific immunity is seen in like granulocytes, basophils, eosinophils, like red blood cells. All of them, you will have no dendritic cells in the brain. Okay, so along with that, mast cells and macrophages, these are all the cells which will show the innate immunity. We know that acquired immunity means we'll acquire this immunity when the host parasite enters into our body. Innate immunity is already present, but it is specific. Cell mediated immunity means which is like what you will say, like cell mediated immunity is nothing but the immunity which is mediated with the help of some cells, okay, according to the infection. And humoral immunity is normally seen in like infections and everything. Okay, if we move on with, so what is the right answer here? You can say that the innate immunity is the immunity which is normally seen in at the time of birth, and it is also known as non-specific type of immunity. Okay, we'll move on to the next question. What is the next question is, it came from the bryophytes itself, from the, okay, in bryophytes the zemme help in which of the following? So we know that the bryophyte plants will have a specific structures that are nothing but zemme. Okay, if you see the picture here, you can see that these are the, like, you can see like bulb like structures, no, dot dot dot, so those are known as zemme here. If you see the picture here, you can see that these are known as gemma cups, in which you can see the zemme. They are like special structures that are present in the bryophytes actually. So what is the role of these zemme? Are normally they are like they are produced by the fragmentation of thalli. Fragmentation means, like, you can see they are fragmented, like divided. If you see that they are fragment like structures, no, draw line by line, line by line. So due to the fragmentation of the thalli, some of the zemme are formed in a cup like structures known as zemme cup. Okay, so what is their role? Means they are, so here they are formed like they are not formed with the help of any parent or something, then means they are formed on their own. So normally what they'll do, if you see that what is their appearance, means they are normally green and multicellular in color, and they are asexual buds. Why? Because these buds are not formed by the sexual reproduction with the like of ovary or sperms or like that. It is asexually formed in the body itself and located on the thalli. Okay, they are located on the thalli of the bryophytes. So what is the main role of these gemme is they'll help in the asexual reproduction. Okay, so asexual reproduction of the bryophytes, that is their role. We'll move on to the next question. What is the next question is, in frog the renal portal system. So this question frame came from the frog structure. Okay, what they are asking means, in frog the renal portal system is a special venous connection that acts to link. So here they are asking what is the role of the renal portal system and which it will connect, which parts of the body of the frog is connected with the renal portal system. If you see the picture here, normally, look, this is the renal portal system. Okay, so normally if you see that this renal portal system is mainly connected to the kidneys of the frog, and it is extended to remaining parts of the like lower parts of the frog and after the kidneys. Okay, remaining parts of the body and the kidney are connected with the renal portal part. So here you can see renal. Okay, so this is the renal vein which will connect the kidneys, and then from kidneys these renal veins are passed on to the remaining parts, means lower parts of the body. Okay, so what is the option here? The renal portal system obviously connects the kidney and the lower parts of the body. Okay, will lower parts like small intestine, large intestine, pelvic region and everything. Okay, we'll move on to the next question. And here you can see that there are two statements given in this question came from the ecology itself. Okay, as I observed that there are so many questions coming came from the ecology in this paper actually. Okay, if we move on to that, they have given the two statements. In the first statement, they have said that in ecosystem there is an unidirectional flow of energy of sun from producers to consumers. Okay, in the second statement, ecosystems are exempted for second law of thermodynamics. In this, you have to say that both of them are true or both of them are false, or either one of them is true. So if you see that for in the first statement in ecosystem there is unidirectional flow of energy. We know that unidirectional flow. Why they are known as unidirectional means here the energy is flowing from producers to consumers. We know that there are prosumers, primary consumers, secondary consumers, tertiary consumers like that. Okay. So here the producers are nothing but the plants. Whereas the primary consumers are that which will eat the plant and everything, and then secondary consumers are that which will feed on the primary consumers, and tertiary consumers are like that, and decomposers. So normally here if you see that the producers are the organisms which will take the energy from the sun, and then they'll pass on that energy to the upper levels of the food chain. So here you can say that it is only unidirectional flow, means the flow of energy is seen in the unidirection only, not they again come into this. Why? Because the animals will not give that type of energy. Okay, normally if they're dead they'll give the nutrients but not the energy. So this is an unidirectional only. So this is right, the statement 1 is right. Whereas if you see the statement 2, they are not exempted for the second law of thermodynamics. Why? Because they need a constant supply of energy to synthesize the molecules, right? Why? Because if uh, first of all the producers need the sunlight to show photosynthesis, and then the producers were like these primary consumers depend on the producers, and secondary will on the primary, like that will happen day by day, every time, every second. So they need the constant supply of energy to synthesize the molecules they require. That's why they need continuous law of thermodynamics, means they need the universal tendency towards the increasing disorderliness. Means if the energy is stopped, flow of energy is stopped, obviously flow of energy is stopped means if the plants does not undergo photosynthesis they'll not grow. So if the plants will not grow, the primary consumers are like they are affected. By because they don't have any plants to eat. So and then the secondary consumers which were depending on the primary consumers will also be affected. Why? Because these animals will die because of the lack of food, like that it will happen. So in the ecosystem, in the food chain, the continuous flow of energy is needed. That's why they are not exempted for the second law of thermodynamics. Okay, we'll move on to the next question. What is the next one is it came from the Calvin cycle actually, like okay, so Rubisco enzyme if you see it. Okay, so Rubisco enzyme is nothing but we know that this is an enzyme which is helpful for the transfer like carboxylation of RUBP, means ribulose bisphosphate. Okay, we know that it will happen normally. This enzyme is also helpful in the photorespiration and photosynthesis stage too. Okay, means it absorbs both oxygen and carbon dioxide. Okay, what it will do, it absorb the carbon dioxide and it is converted into waste product, whereas the CO2 is converted into energy. Here means Rubisco is the enzyme which can both show both photorespiration and photosynthesis. If you see here, you have to say which is true about Rubisco. First statement is it is active only in the dark. You cannot say that it is active only in the dark. Why? Because it is active also in the light. That's why it is showing the photorespiration and photosynthesis. No, so this first option is wrong. It is not correct, and it has higher affinity for oxygen than carbon dioxide. Actually you cannot say that it is having higher affinity. If it is having the higher affinity of oxygen, it will not fix the carbon dioxide in the photosynthesis stage. It will be included in the respiration process. So this is not correct, means it is not having higher affinity. Okay, and it is an enzyme involved in photolysis of water. They are saying no, no. Photolysis of water normally takes place in the light reaction, right? In the light reaction itself, the Rubisco enzyme is not involved. Photosystems are involved. So this enzyme is not involved in the photolysis of water. If we move on to the last option, that is nothing but it catalyzes the carboxylation of RUBP. We know that. What will happen in the carboxylation of RUBP means if you see the cycle, the Calvin cycle, there are three stages like carboxylation, reduction and regeneration. So in the carboxylation means what it will do, it will convert these RUBP ribulose bisphosphate molecules to phosphoglyceric acid molecules with the help of carbon dioxide. Okay, so this Rubisco enzyme mainly plays role in the carboxylation of RUBP to phosphoglyceric acid. So this is the main role of the Rubisco, and it is true. This statement is true that it catalyzes the carboxylation of RUBP. Okay, we'll move on to the next question. What is the next question is, which of the following enzymes are not essential for gene cloning? So here they have given so many enzymes, in that you have to say which enzymes are not involved in the cloning of genes. Okay, if we see that there are so many enzymes, restriction enzymes, DNA ligase, DNA mutase, DNA recombinase and DNA polymerase. So in this you have to say which is not involved. Okay, not essential for the cloning. Okay, if you see that cloning means what? Producing the same like more amount of copies of DNA. Or you can say if we are taking here DNA, so you can say that DNA. Okay, more upon if you see the gene cloning here, what we'll do first of all in the lab, they'll take any cloning vector like a plasmid or something, and then they'll take the desired DNA. For example, if they want to produce more amount of DNA that is responsible for some eye color eye color of the like some white. Okay, what it will do, if they want that specific gene, what they'll do, they'll take the gene which is coding the DNA, which is coding for the eye color of the child. So what they'll do, they'll take that eye color coding like DNA, and then what they'll do, and in the next step, first of all in the first step they'll take the cloning vector or the plasmid, and they'll cut that vector at specific site so that they can insert the new DNA. So this cutting is done with the help of the restriction endonucleases, we know that. So along with that, here also the restriction endonucleases will play main role in cutting the desired DNA from the eukaryotic chromosome, eukaryotic or prokaryotic. Okay, so what will happen, these desired DNA molecules are then attached to the plasmid of the host. Okay, like taken from the host. So that for attachment of this DNA to this plasmid, we need a DNA ligase enzyme. So here second enzyme that is participating in DNA cloning is DNA ligase. Whereas what happened in the next step means these plasmids which were having the desired DNA were inserted into the host cell. It may be a bacterium or anything. So when they are inserted into the host cell, so what they'll do, they'll add some enzymes that are nothing but DNA polymerase enzymes. Polymerase means what? Making of copies, right? So what they'll do here, those desired DNA containing host cells will make so many copies with the help of the DNA polymerase enzymes that are present. Okay. So here what were the main enzymes that were normally the DNA polymerase is also plays main role here. Why? Because they'll take the single strand of DNA from the required chromosome, and then they'll make the copies of this DNA. So many copies. They'll not take only one plasmid. No, they'll take so many plasmids. So to produce the so many copies of the desired DNA, they need the DNA polymerases. Okay. So what were the enzymes that were included in the DNA cloning? Or first one, restriction endonucleases, next DNA polymerases, and then DNA ligases. So according to this question, what were not involved are, here you can see that restriction enzymes are involved. Okay, DNA ligase for the attachment of required desired DNA to the plasmid, and DNA mutase. Actually DNA mutase is the enzyme which will help in the mutation of the genes. Mutation of the DNA means sudden changes, so it is not involved in the DNA cloning. Whereas DNA recombinase is an enzyme which will show the recombination of DNA, like it is normally used in the meiotic divisions or like that. Okay, and DNA polymerase is the enzyme which is helpful for making the copies of DNA. So here A, B and C are the enzymes which are normally essential. So C and D are non-essential. So you have to pick the answer. C and D are the enzymes that are not essential for the gene cloning. Okay, we'll move on to the next question. This question came from the plant growth and development. You have to read the statements, and they have given it them from the hormones, plant growth hormones. Okay, you have to say whether that is correct or not. In the first one, they are saying that parthenocarpy can be induced by auxins. We know that the parthenocarpy can be induced by the auxins. Okay, parthenocarpy means what? Formation of the seedless fruits. Okay, auxins will help in the production, seedless fruit production, right? So this is the right statement. Whereas the plant growth regulators can be involved in promotion as well as inhibition, right? This is right. Why? Because what they'll do, if the plant is having all the promoters like auxins, gibberellin, cytokinins, abscisic acid, everything, they'll each hormone is having the specific role to play in a specific part of the body. If what they'll do means, if the auxins are enough for the production of shoots or something, if the shoot is developed at a certain extent, what they'll do, the production of auxins is stopped, means they'll not play a major role in shoot elongation. Why? Because the shoot have to maintain at certain height. So what they'll do, they'll like the role of the auxins is decreased, means like it will not promote like outer growth of the shoot. So like that, the normally any hormone or the plant growth regulator will is involved in the promotion as well as inhibition of the growth. Okay, according to the in their production. Okay, we know, simply we can say that if anything, if for example you have food in your home, you can eat the food which is sufficient for you, means when you feel that your stomach is full, you will not eat, right? If you feel like if you eat more, what will happen, your stomach will upset, and you will have any vomiting sensation or like that, means you will suffer. Like that only, when the auxins are needed, then only they are produced. If they are produced in the more amount also, what will happen, it will show the reverse reaction. So that's why they are also involved in promotion and inhibition of growth. Okay, this is also right about the plant growth regulators. If we move on to the third option, de-differentiation is a prerequisite for redifferentiation. De-differentiation matlab differentiation ko stop karna, means you are stopping the differentiation of the cells. Whereas the redifferentiation will occur when the de-differentiation is stopped and the cells will grow, and then they redifferentiate into different types of cells. So it is also a prerequisite method. Okay, this is also right. And fourth option, abscisic acid is a plant growth promoter. No, it is not a plant growth promoter actually. It is a plant growth inhibitor. Okay. And apical dominance promotes the growth of lateral buds. Normally, apical dominance will not promote the growth of like what they'll say lateral buds. It will inhibit the growth of lateral buds. Okay, so here D and E are wrong, whereas A, B, C and the correct options. In this, the A, B, C only are the correct options. So option 1 is correct. Okay, we'll move on to the next question. That is from, it is about the termination of transcription process. Here they are asking which factor is important for the termination of transcription. Here they have given the options alpha, sigma, rho and gamma. In these factors, which one is responsible, means normally we know that rho dependent termination will occur in the prokaryotes, right? What will happen means this rho factor, which we know that what it will do, it will attach to the mRNA that is synthesized from the DNA. Okay, that was synthesizing from the DNA. Here you can see two strands. In this, from the one strand only mRNA synthesized. No. So what will happen means, when the transcription have to stop, means that the mRNA or the DNA thought that okay, this much of mRNA is enough, then it have to stop, means the row factors which are present in the cytoplasm or like in the ribosomes, what they'll do, they'll be attached to the mRNA which was synthesizing which is synthesizing from this DNA. And when they are attached to the mRNA, they'll move like this. So when they are moving like that, what they'll do, when they'll come to this extent, like when they'll settle here, what they'll do, they'll remove this yellow part is nothing but the RNA polymerase. So when they are attached to this RNA polymerase, they'll stop the action of the RNA polymerase by removing it from the RNA. Okay, so that you can see that evolution of mRNA strand, and like this RNA polymerase is separated, and you can see the DNA double strand here. Okay, like that, this mRNA, this RNA polymerase, and here, and the rho factor is attached here. So like that, the rho factor, rho dependent termination will occur in the transcription. Okay, here you can say that the rho factor is the important one for the termination of the RNA. Okay, we'll move on to the next question. What is the next question means? It also came from the frogs itself. Okay, and what is that means? Here they are asking that frog respire in water by skin and buccal cavity, whereas on land by skin, buccal cavity and lungs. So here they have said that in the question that frogs will respire in the water with the help of the skin and buccal cavity, and on the land by skin, buccal cavity and lungs. If you see the picture here, you can see that the total structure of the frog. Like if you here you can see that this is the skin. We know that normally the frog, when it is in the water, it will respire, means it will take the air inside with the help of the skin actually. Okay, so buccal cavity is present here. Normally it will not open the mouth, no, so the buccal cavity is not responded. We have to respond, means it have to open its mouth and then respire. So the buccal cavity is not responsible for the respiration in water. But when it comes out of the water, what it will do, it will open its mouth, so that the buccal cavity which is present in the mouth, through that it can respire. Okay, so here you can say that the frogs will respire in water only by the skin, not by the buccal cavity. Okay, so here you can say that the statement, they have given the options that the statement is true for water but false for land. It is not true for water. Why? Because the buccal cavity is not involved in respiration in water. So this is wrong. The statement is true for both the environment. No, no, it is not true. And the statement is false for water but true for land, obviously. In the land, when it comes, means what they will do, the skin, lungs and buccal cavity, three of them will play a main role in the respiration for the frog on the land. So here what is the correct option is, the statement is false for water. Why? Because the frog will not respire with the help of buccal cavity in the water. Okay, so it is false for water but true for land. Okay, we'll move on to the next question. What is next question is, here they have said about the twins. Okay, monozygotic, dizygotic twins, we know that. Here they have asked the question from the twins. They are saying that twins are born to a family that lives next door to you. Okay, they are saying that when you are having the neighbors which were next door to you have the twins. Okay, the twins are normally a boy and a girl. So the twins which were born to your neighbors are boy and a girl, and you have to say which one of the following must be true in these statements according to the question. So here they are saying that if they are boy and girl, normally we know that there are so many types of twins, like identical twins, fraternal twins, and in that also monozygotic twins, dizygotic twins are there. Monozygotic twins means which will have the, what they'll say, simply same zygote, means either two females or two males or nothing but, like these are identical. Monozygotic twins are like two females or two males or like that. Okay, so whereas identical means they'll have maximum similarity, means 75 to 80% genetical identity will be there. Whereas the fraternal twins are nothing but they may be boy and a girl, or a girl and a girl, or boy and boy. So fraternal means they'll not show the maximum similarity. Okay, around 50% of the similarity will be seen in these fraternal combinations. If you see here, these two boys, one is height and one is low, and face is also different, and that in these two girls also it will happen. And if you see here, girl and boy are also different from the fraternal, and these are also known as dizygotic twins. Okay, so according to this, what is the correct answer is they are monozygotic twins. No, 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 they are not monozygotic twins. Why? Because monozygotic means here the boy and girl are born. No, they are not monozygotic. So option 1 is wrong. Whereas they are fraternal twins. Obviously, the fraternal twins is a right answer. Why? Because they are not, they are boy and girl, and they are showing the similarity. Okay, they are the boy and girl. And another thing is normally boy and girl means nothing but they are fraternal twins only. Means boy and girl cannot be born in the identical twins. They are known as identical twins when they'll have the similarity in their genes. Means in the chromosomes, all the chromosomes like allosomes and autosomes. Here, identical twins are normally seen in boy and girl and girl, boy and boy, girl and girl, but not boy and girl. Boy and girl twins are known as fraternal twins. Okay, so these are fraternal twins. If you move on to the third option, they were conceived through in vitro. No, no, they are not conceived through in vitro fertilization. Normally, the fraternal twins can be conceived normally. Okay, and they have 75%. No, no, you cannot say that 75%. Why? Because here you will have the dizygotic twins. They are not that much similar. Okay, so that's why this option is also wrong. In this, you have to say that the boy and girl which were born to your neighbors are fraternal twins. Okay, and next question is from microbes. Okay, which of the following microbes is not involved in the preparation of household products, they are asking. So we know that there are so many types of microorganisms which are involved in the household, like we can see in the normal daily life. So if you see here, what is that? First one, Aspergillus niger. So Aspergillus niger is a main role. Normally what it will do, it plays main role in the production of citric acid. Okay, means citric acid production is mainly in the citric acid production in the house or household food items, we'll use the Aspergillus niger bacteria. Means normally not we use, normally the chefs and everything they'll use, like in the bakeries also they'll use the Aspergillus niger bacteria powder or like that to for the citric acid production. And next one, lactobacillus. Lactobacillus is a common known bacteria. Why? Because it helps in the formation of curd from the milk, right? So lactobacillus is also household bacteria, whereas the trichoderma polysporum, it is not a household. We'll discuss it what it is used for. Whereas Saccharomyces cerevisiae is used in the bakeries. Okay, for bakeries for yeast production. Nothing but it is an yeast. Saccharomyces cerevisiae is nothing but a yeast. So it is mainly used in bakeries and cake and everything. Whereas the Propionibacterium shermani is also used in production of cheese. Okay, in the cheese industries or in the production of the cheese also we can use the powder of that sample of this Propionibacterium shermani. Okay, so here normally the household bacteria are nothing but here you can say that Aspergillus niger, Lactobacillus, Saccharomyces and Propionibacterium. So whereas if we see the Trichoderma polysporum. So what is its main role is, so this Trichoderma polysporum is normally known as a biocontrol agent. Means what it will do, it will control the pathogens that were affecting the growth of the plant. Means it will help in the plant defensive system. Means it will help the plant from the pathogens which are present in the soil, which like the parasites or pathogens which want to like infect the plants or everything. So these trichoderma or the bacteria normally what they'll do, they are present in the soil, they'll help in the defensive system of the plants. Okay, so like that they are not used in the household product, and they are also known as biocontrol agents. And sometimes these the trichoderma polysporum is used for the immunosuppressive drugs like cyclosporin. Okay, we'll move on to the next question. What is the next question here is, here they have given the questions like hormones, and you have to say that these the places where these hormones are produced. Okay, so if you see the first list, they have given the hormone. Second list, they are places where they are produced. Okay, if you excuse me, so progesterone, we know that the progesterone is a female sexual hormone. Normally it is present, it is produced from the corpus luteum, okay, of the female reproductive system. Whereas the relaxin, what is the role of progesterone means it is a sexual hormone, okay, it helps in the sexual reproduction of females. Whereas the relaxin is the hormone which is also present in the females, and it is produced from the ovary. So what is the role of this relaxin is it helps in the birth of the child. Okay, when the means what it will do means it will relax the muscles of the ovary when the birth, when the child have to born. What they'll do, if we what we'll do, we'll get more pressure now when the child have to come out, so it will relax some of the muscles so that the child can be easily born. Okay, and another thing is melanocyte stimulating hormone. Melanocyte means we know that the melanins are the skin pigments, like the pigments which will help in the color of the skin. Okay, so this is normally present, produced in the pars intermedia. Okay, and catecholamines are the hormones which are normally produced from the adrenal medulla. What is their main role? Catecholamines, they'll help in the, like, they are normally, there are so many types like epinephrine, norepinephrine, or noradrenaline, or adrenaline, non-adrenaline. These are all known as catecholamines. Okay, they are normally, they produce, they are produced from the adrenal medulla, okay, which is present in the kidneys. Okay, so here if you match it, A is nothing but 4. So here options which are having the A 4 or 2, and B is nothing but 2. In this, these two are also having B2. And melanocyte stimulating hormone C is nothing but 1. So the first one is showing the C1 here, and catecholamines D is nothing but 3. So here the first option A4, B2, C1 and D3, this is the correct option in which the hormones are produced. Okay, we'll move on to the next one. What is the next question is, here they have given the question from the staining, like the bacterial colonies and everything. Actually we have discussed this question in the PYQ session of our before NEET exam. Okay, so what is the question is, they have asked that the blue and white selectable markers, the blue and white selectable markers have been developed which differentiate recombinant colonies from non-recombinant colonies on the basis of their ability to produce color in the presence of chromogenic substrate. Okay, so in this, they are saying that two colonies are there. One is blue and another one is not blue. Means one colony is producing the blue color colonies and another one is colorless. According to that, you have to say that the blue colony is producing, like, the colonies which are producing the blue color are known as recombinant colonies, and the which are not producing the blue color are non-recombinant colonies. Okay, on the ability of their producing colors in the presence of chromogenic substrate. Here they have given you two statements. First one is the blue colored colonies have DNA insert in the plasmid and they are identified as recombinant colonies. Okay, the second one is the colonies without blue color, means they may be colorless, have DNA insert in the plasmid and are known as identified as recombinant colonies. Okay, if you see here what they are saying, that blue and white selectable markers. Okay, selectable markers means here, like we have discussed in the lag Z gene, normally we know that beta galactosidase enzyme process, when the DNA is inserted into the beta galactosidase enzyme, like the marker place of the E. coli bacteria plasmid, what happened actually, when the DNA is inserted into it, the blue colonies were turned colorless, like that, no? So according to that, like that question we have revised in the PYQs. So here we have to see it is included with the laxedzine. In that you have to say which statement is correct, like both of them are correct or both of them are incorrect, first one is correct or second one is incorrect. Okay, if we see, so Normally, if we see here in the structure, you can see that they have taken the plasmid vector and which they have induced the foreign DNA. Here we are discussing it in the lag Z gene. Okay, so when these they have inserted in three ways. First of all, they have inserted that foreign DNA into lag Z gene, and they have in the second colonies they have inserted the lag Z gene outside the insert outside the lag Z gene, and third they did not inserted the colonies. So what was the result means these plasmids were again inserted into the E. coli bacterium. What happened means some of the colonies have shown the white colonies means the genes, the inserts which are present inside the lag Z gene have shown the white colonies, and which are present outside the lag Z gene the inserted are showing blue colonies, and without not having any insert are also showing white colonies. Okay, here what they have said that the blue white screening is based on the disruption of the laxedzine, right? Why? Because presence of the laxedzine is showing major impact on the coloring of the colonies. Okay, if your plasmid contains, for example, if the plasmid contains a insert, okay, laxedzine is disrupted and white colonies are formed. Means if we insert into the laxedzine, the laxedzine cannot show its action, so that the color of that laxedzine is inhibited, so that white colonies are seen. Okay, laxedzine normally shows the blue color, where whenever that laxedzine is suppressed, the DNA is more it will suppress the laxedzine, it will show more efficiency, then the white colonies are formed. Okay, and they are recombinant. Why? Because the recombined genes are present here, means laxed and the DNA are recombined with each other. In the DNA showed the dominance and it is showing the white colonies. Okay, and if the plasmid does not contain an insert, laxed is intact. Okay, for example, it does not contain any insert, so it is intact and shown the blue genes, and the colonies turn blue and non-recombinant, but not recombinant. Why? Because here the DNA is not recombined with the laxed gene. So here, according to the question, you can say that the statement 1 is correct and 2 is wrong. Means option 3 is right from this question. Why? Because in the statement 1 they have said that the colonies produced were like white colored and they are recombinant. In the statement 2 they have said that they are also recombinant, but they are not recombinant. They are colorless and recombinant. They are not recombinant. Why? Because colorless means they are not, they are suppressing the laxed gene means, okay, then they are nothing but recombinant. When they are not suppressing the laxed gene means they are non-recombinant. Okay, so here statement 1 is correct and statement 2 is wrong. If we move on to the here in the picture, you can see that some of them are blue and some of them were white. So these blue colonies are having the laxed genes, and these white colonies are having laxed genes, but that their action is suppressed by the DNA inserts into them. Okay, if we move on to the next question, and nothing but the last question from this paper, like from the biology session of that paper code 45 from the paper NEET paper 2025. So here they have given these question from the equation, here they have given some equations. You have to say which one is Verhulst Pearl logistic growth of population. So according to the ecology, they have given so many population growth curves like sigmoid curve, J curve, etc. Like we discussed this question also in the ecology PYQs. So we will according to the curve formation only, the Verhulst Pearl, they have created a logistic growth equation for the population. So according to this equation, it describes the population growth that slows at the as the population reaches the carrying capacity. Here you can see, if you see here, what is k is nothing but the carrying capacity, n is number of populations, t is time. Okay, so means when the population will grow for to certain extent, when obviously the resources are less and they are limited, obviously they'll show the carrying capacity of the population is decreased or maintained in a certain manner. So that is nothing but the Verhulst Pearl logistic growth of population. So according to this, you can say that n is population size, r is nothing but the intrinsic rate of natural increase. Okay, natural increase means to the natural evolution, and K is carrying capacity. So dn by dt means rate of change of population side. So here according to this, the dn by dt, the rate of change of population size is equal to the rate of carrying capacity, then the number of population density minus the carrying capacity. Okay, here what is the correct statement is dn by dt is equal to Rn into k minus n by k. So here in this question the option 2 is correct. So in this, by this equation you can see the carrying capacity or the population, the density, the number of population changes according to the environmental factors or the limited resources. Okay, so that's it guys. So this is all the questions that were came from the biology session of the paper 2025 from paper code 45. Okay, so I think that I have revised all the questions, and I think that all of you understood the questions like I have explained deeply. So if we move on that so the paper revision was over, so we are stopping it here, and in the next videos we are going to start a neat crash course, so that you can start from the long term, like if for example if you are a class 11 students, like if you want to uh, admit or not admit, sorry, like if you want to write the NEET 2026 exam, like if you are class 11, you can see like join my NEET crash course in the Biosastra Talks. If you want to join the classes, you can contact me through my mail or the phone number I have given in my channel. Okay, and if you want to contact me or if you want to join the crash course, you can comment me in the comment section in the Biosastra Talks channel, or either you can contact me through the mail or the phone number I have given. Okay, so, thank you, and I'm hoping that you want to join the crash course. And again, another thing, if you are not, we are also class 12 students and who want to do the NEET long term, okay, you can also join along with me in the Biosastra talk, so that we can continue the crash course, and you can go on with the revision of each and every topic along with some PYQs and everything through this crash course. Okay, thank you guys. I hope all of you understood the topics I have explained till now. So I am hoping that you'll stay on with me. And if anyone want to contact me, you can contact me through my mails or through my channel. And that's it. And if you like my videos, just share it with your friends so that they can also be helpful. And if you want to stay on with me, or if you want more videos from my channel, you can go on with the, sorry, you can subscribe my channel Biosastra Talks. Okay. Thank you guys.